Okay, I'd like to invite our next speaker up. So this is Zhang from Southwest University, who's going to talk today about functional gradients of the striatum and its linkage with frontal cortical function in cocaine addiction.
Hello, everyone. My name is Zhang. I'm a PhD student at Southwest University in China. Thanks for OHBM for this opportunity to share my recent research with you. Today, I will discuss functional gradients of striatum and its linkage with frontal cortical function in cocaine addiction. Well, the striatum plays a critical role in the development and maintenance of CUD. It's mirrored as its segregated circuits and heterogeneous functional organizations. The ventral stratum associated with reward processing and impulsive behavior receive inputs from the limbic end of the frontal areas. In contrast, the dorsal stratum linked to the habit formation and compulsive behavior receive projections from the associative and the motor areas. Neuroimaging studies indicated the repeated cocaine use impairs the structure and functional connectivity in executive, reward, and stress-related circuits. Additionally, transmission from the impulsive driven to compulsive cocaine use is mediated by a shift from the ventral to dorsal striatal control over behaviors. Despite these insights, most previous studies used ROI analysis, potentially overlooking striatum's complex and overlapping representations. As shown in this figure, another study highlights distinct pattern of altered ventral stratum. Another study highlights the distinct patterns of altered ventral stratum connectivity in CUD missed by the whole ventral stratum analysis, emphasizing a need for approaches that model striatal topography's continuous nature to characterize connectivity alterations better. Well, actually, recent studies used gradient-based analysis to reveal striatal function is organized along overlapping and continuous gradients, with first mode representing ventral medial to dorsolateral gradient associated with cortical striatal mediated goal-directed behaviors, offering a novel direction for understanding striatal function in CUD. Indeed, gradient-based analysis have emerged as a powerful tool for capturing abnormalities in topography of connectomes in patients because they offer comprehensive framework for characterizing functional modulus and reflects hierarchical organization of brain functions with high reproducibility and reliability. For instance, several studies use these approaches to reveal altered microscale gradients in ASD and MDD, as well as abnormal single regional gradients in schizophrenia and psychosis. However, the striatal functional gradient in CUD has not been investigated. So to address this issue, the primary object of our study is to investigate alterations in functional gradient of striatum in CUD using collectivity gradient analysis, particularly focusing on ventral medial to dorsolateral gradient. Okay, in our work, we use resting state fMRI data from publicly available Mexico MRI datasets with 41 CUD patients and 44 healthy controls. These 55 subjects completed fMRI scans, clinical questionnaires, and provided tobacco use informations. To validate our findings, we used NIDA datasets with 53 no treatment thinking cocaine users and 45 healthy controls. After standard pre-processing with fMRI prep, we conducted collectivity gradient analysis. In this analysis, we constructed individual FC metrics at voxel levels using striatal mask from Harvard Voxelfold Atlas. For each subject, whole brain FC matrix was mapped for each voxel of striatum. We then computed coincidence-based similarity metrics and applied diffusion map abandoning to generate gradient component that account for collectivity pattern variance. This results continuous gradient maps representing voxel connectivity similarities. Well, to compare the HC and the CUD group, we use a group level gradient component template derived from averaged FC matrix from an independent healthy control cohort. The observed ventromedial to dorsolateral gradient of stratum explained the second largest variance and similar with previous studies. Well, here are mean ventromedial to dorsolateral gradient in two groups. A visual inspection of the global histogram reveals there is a prominent expression of stratum's lowest and highest portions. Additionally, significant comparisons between two groups revealed significant difference in gradient range and gradient variance, indicating the more differentiated collectivity patterns in CUD. Furthermore, we found significant gradient changes in ventral and dorsal stratum, specifically increased dark red voxels in nucleus accumbens and ventromedial caudate, as well as increased dark blue voxels in dorsal medial caudate, suggesting the neuroplastic changes induced by the repeated cocaine use. Our meta-analysis further linked these changes to disruptions in goal-directed behaviors, including reward and motivation-related cognitive functions. Notably, in the validation datasets, we replicated the gradient alterations in ventral stratum, but not the dorsal stratum. This may be explained by the low drug dependence in cocaine users in validation cohort compared to the CUD patients in main cohort. Well, following that, we conducted partial correlation analysis to understand the relationship between gradient alterations and clinical characteristics in CUD. Actually, we found gradient variance significant correlated with drug-dependent severity. Moreover, cocaine intake was associated with gradient values in both dorsal and ventral stratum. These findings confirms that the striatal circuit alterations are evident across addiction continuum, needing survey dysfunctions in cognitive control and reward processing. Well, next, we use these region-based functional collectivity patterns to understand how gradient alterations link to the whole brain collectivity. Consistent with previous studies, we found the dysregulations in striatal frontal circuit in CUD patients, particularly the decreased functional collectivity between ventral stratum and left DLPFC. Well, next part, given previous studies suggested the rTMS targeting the DLPFC have strong effect on cocaine craving and impulsivity, as well as the change the abnormal brain circuit. We use the SUD-max TMS datasets to test whether the rTMS on left DLPFC could modulate the gradient values in CUD patients. Well, in these datasets, two groups of CUD patients assigned randomly received either active or sham rTMS on left DLPFC and underwent pre- and post-treatment assessment. A significant group-time interaction effect was observed in the ventral stratum but not dorsal stratum. The post-hoc test further revealed the rTMS significant lower gradient values in active group compared to the sham group in ventral stratum. This finding suggests that rTMS targeting the DLPFC could change the abnormal gradient values in CUD patients. In conclusion, our study provides novel evidence of striatal functional gradient dysfunctions in CUD, links the dependent severity and the cocaine use. Well, the abnormal striatal-frontal circuit may drive this disrupted disrupted gradient mood. Moreover, rTMS targeting the left DLPFC shows promise in modulating these gradients, offering potential therapeutic approaches for CUD. Well, finally, I extend deepest gratitude to Prof. He, Prof. Yang, all collaborators and researchers who contributing to data sharing. Your support and collaborations have been invaluable. Thanks for your attention.
Hi, thank you, Ran. We have one question online. How do you explain rTMS targeting the dorsolateral prefrontal cortex modulates the abnormal gradient in ventral striatum but not dorsal striatum? And
Okay, thanks for your questions. Actually, I remember as shown the previous presented figures, the ventral medial cortex involving in ventral medial areas will many show the collectivity with the DLPFC, right? While the dorsal striatum many correlated with the motor areas. So we think this could be explained why the stimulation on the DLPFC just change the gradient values in ventral striatum. Yes,
Thank you.
thanks.
One last question. Did you control the multi-drug users for your study?
What?
Yeah, did you control for the, I'm guessing so it says, did you control for the multi-drug users in your study? I mean, you showed tobacco, for example, as a covariate, and I know there were some other drug use in those studies. Did you control for that or not? That's just a quick question.
You mean I control other variables in
other
our
like
study?
drug use, because usually patients use more than one drug. I mean, this was cocaine specifically, but there were other drugs like tobacco, and I think you control for that.
Yeah, actually, I think because you know the Mexico data set, they provide some information on it, and we exclude some participants label has some depression or other psychiatrists. Maybe some use some drugs, but I didn't find any information about the other drug use in these patients. But in the NIDA data sets, I'm sure they didn't use other drugs during this experiments. Yeah, thanks.
Okay. No, it's good. We can talk later about that. I'm the one doing this. So.
Yes, very thanks.
Great, thank you. Do we have more? Yeah, is there any other questions? We have one more space. Otherwise, thank you. Thank you for the talk.